Hello, everyone. I'm Dunja Mladenovic, General Relations Specialist from the MDPI, and I will be your host for today. Welcome to the webinar Announcements in Food Nutrition and Bioactive Compounds, organized by MDPI and the Journal of Applied Sciences. Before introducing the MDPI journal and the lineup of today's event, I would like to remind everyone that this webinar is being recorded and the recording would be made available as soon as possible on the webinar's webpage. Please note that you need to be logged into the site forum to see the recordings. For the beginning, let me introduce you with the MDPI. MDPI is a pioneer in scholarly open access publishing, and it has supported academic community since 1996. It's based in Basel, Switzerland. MDPI has the mission to foster open scientific exchange in all forms across all disciplines. Our 484 diverse and open access journals, including 477 peer-reviewed journals and 9 conference journals, are actually supported by more than 295,000 academic experts who share our missions, values, and commitment to providing high-quality services for our authors. Now, Applied Sciences is an international peer-reviewed open access journal on all aspects of applied natural sciences, published semi-monthly online by MDPI. It publishes reviews, research papers, and communications. Our aim is to encourage scientists to publish their experimental and theoretical results in as much details as possible. Today, there would be 3 presentations, 20 minutes each. At the beginning, our chair, Dr. Danila Kanciossi, will open this webinar and will introduce each speaker. There are some slight changes in the program, so Dr. Maria Verilo would start this webinar. Then Dr. Yasmani Armaz Diaz would follow up, and after him, Dr. Davide Raffaelli. Once they're all finished with presentations, there would be a Q&A session moderated by our webinar chair, Dr. Danila Kanciossi. Additionally, this webinar is actually related to the special issue in Applied Sciences named Advancements in Food Nutrition and Bioactive Compounds, guest edited by our chair, Dr. Danila Kanciossi. This special issue belongs to the section of Food Science and Technology, and it will be open for submissions until the 20th of April 2026. Please note that this special issue is dedicated to exploring the latest innovations and scientific discoveries regarding the role of nutrition and bioactive compounds in food, with the aim of deepening our understanding of their impact on human health. If you are interested in finding out more details and potentially submit your manuscript here, please feel free to check the special issue website. During this webinar, if you have any questions for our speakers, please feel free to post them at any time in the Q&A box. You can find it in the Zoom menu bar at the bottom of your screen. It would be great if you could use Q&A option and not chat panel for the questions for the speakers. And now for the end, let me introduce you with today's webinar chair, Dr. Danila Kanciossi. Dr. Kanciossi holds master's degree in applied biology and earned her PhD in biomedical science at University Politecnica della Marche, where she investigated the effects of Manuka honey on colorectal cancer stem-like cells. She has completed multiple postdoctoral fellowships, including 2 awarded by Fondazione Umberto Veronesi. Focusing on the interplay between obesity, bioactive compounds, and breast cancer biology. She also serves as adjunct professor for the course of food composition tables, diet planning for athletes in the first level master's program in nutrition and applied dietetics in sports. Her scientific expertise lies in the use of advanced in vitro models, including 3D cultures and co-culture systems to explore the therapeutical potential of food-derived bioactives. She is also author and co-author over 70 peer-reviewed articles and more than 20 conference abstracts. Dr. Kanciossi actively contributes to as editorial board member and reviewer for several journals and has served as guest editor and topic editor in multiple special issues. Dr. Cacciosi, thank you very much for chairing this webinar. That would be all from me. I'm handing over the floor to our chair. Enjoy, everyone.
Good morning, everyone, and welcome to the webinar Advancement in Food Nutrition and Bioactive Compounds. First of all, a warm thank you to Applied Science for organizing and hosting today's event and for providing this platform to share and discuss cutting-edge research. I would also like to thank our three speakers for joining us and sharing their expertise, and thank of all of you in the audience for taking the time to participate. Today, we will explore three complementary perspectives on how natural research and agri-food byproducts can be transformed into valuable tools for human health and sustainability. First, we will discover how compost teas can act as an emerging antiviral and antimicrobial agents, offering an innovative example of circular bioeconomy and green healthcare. Then, we will see how extracts from purple prickly pear peel are being studied for their effect on colorectal cancer cell model, opening new opportunities for the valorization of agro-waste. Finally, we will look at how water management strategies can influence not only the nutritional quality and bioactive compound of strawberries, but also their overall yield and production. Together, these talks highlight the intersection of food science, biotechnology, and the circular bioeconomy, showing how innovation in food research can benefit both human health and the environment. They also remind us that food-derived bioactive compound and the intelligent reuse of agricultural byproducts are not only a path towards sustainable value chains, but also a powerful means to protect and improve human health, from supporting disease prevention to enhancing overall well-being. By linking fundamental research with practical application, these studies illustrate how multidisciplinary approaches can help tackle some of today's most urgent challenges, safeguarding our health, reducing waste, and promoting a greener and more resilient economy. Before we begin, just a quick note, you are welcome to write your question at any time in the Q&A panel. Normally, we collect them and answer as many as possible in the final Q&A section. However, due to family reason, Dr. Verrillo will not be able to stay until the end, so the Q&A session for her will take place immediately after her presentation. Well, let's get started and move into the heart of today's session with our first presentation. Dr. Verrillo is a postdoctoral researcher at the Università degli Studi di Napoli Federico II in Italy, focusing on the extraction and the chemical characterization of compost teas from agri-food residues. Her talk will introduce compost teas as an emerging antiviral and antimicrobial agents and explain how this plant waste-derived extract can support both green healthcare and the circular bioeconomy. Dr. Verrillo, please start your presentation, and thank you.
Okay, the topic of this talk is the application of compost tea as a natural bioactive solution and exploring the antiviral and the bacterial potential of bell pepper and citrus wastes. Innovative and sustainable antibacterial and antiviral solution derived from organic waste offer a promising way to take a health emergency where product with circular economy principle, because this compound has a rich of a bioactive compound, for example, phenolic molecules, flavonoid, protein, and vitamin. And this compound play an important biological activity, for example, reduce the microbial proliferation and reduce the viral replication. This compound use a long application in different fields, for example, cosmetic industries, food industries, and healthcare pharmaceutical application, because they are rich of these biological molecules. Bio-based alternative to conventional drug using a natural compound from organic waste, you offering a cost-effective and friendly solution that reduces resistant risk and environmental impact, because organic waste is important to extractive biological activity, and this waste is not only used for the waste but used as a research, because from this waste is possible extract biological molecules that deployed in different fields. But what is compost tea? Compost tea is a liquid extract produced through the fermentation of compost organic waste and used as emergent and promised candidates to reduce the proliferation of toxic microbial strain, but generally used in agricultural field. But what is the application in medical or cosmetical field? The compost tea solution and the mixture of organic aqueous solution that contain bioactive molecules that can employed in different fields, reducing the microbial proliferation. What is the objective of this work? The presented study aims to assess the antiviral efficacy of compost tea extracted from different resources, bell pepper and citrus wastes, against selected human virus and human microbial strain. The finding made them create an innovative eco-friendly solution that supports circular economy principle and sustainable earth practice. Citrus wastes, such as pear, are known to contain flavonoid and essential oil with antibacterial and antiviral potency. At the same time, bell pepper waste is rich of important molecules, that is caspanoid and phenolic cobalt, that has the antibacterial activity, because unlocking the destruction of membranes and inhibition of microbial replication. During the composting process, the bioactive molecules are transformed and beneficial microbial community, enhancing the power of compost tea to improve the microbial infection. Material method for this work is the composting and the compost tea preparation, because we start to starch material, bell pepper, horticultural resins, and limoncello food wastes, and exploring the composition setup. The composition phase during 100 days, and the extraction is to start to 200 grams only in 1 liter of water, and after several days, solution with freezer dried. The characterization with organic compound will be performed by using the advanced technique, for example, NMR spectroscopy and pyrolysis by CMS assay. Then, when we obtain this extract, we perform the antioxidant activity applied to different spectrophotometric assay, ABTS assay, DPH assay for INSOCAL2 or FRAC, and then we evaluated the antimicrobial activity using two different bacteria strains, gram-positive and gram-negative bacteria strain involved in common human disease. And applied the two different techniques, diffusion disk and broad beam microdeletion, to conference the application of this material. So the innovative of this work is antiviral activity use plaque reduction assay. The test virus is ASV1, RSV1, and poliovirus PV1, and the host cells is verocells 76. The treatment condition is a co-treatment, cell with pretreatment, and virus pretreatment, because we test the molecular mechanism of this activity of these biological molecules. So in this slide, I present the first slide of a characterization by an NMR technique of this extract. In this slide, we use the different composition of this material, and we show that the compost tea extracted by bell pepper has an higher anti-hydrophobic index compared to citrus, and at the same time a lower lean-rich ratio compared to bell pepper. And so it indicates that this compost is a lot of degraded lignin fragment. So we perform and co-validate data using pyrolysis by ZCMS. Citrus compost tea has a greater portion of fatty acid methyl ester, while bell peppers containing the more nitrogen molecules, like derived from microbial activity. These factors support the idea that the compost tea obtained by pepper is a higher microbial contribution, potentially enhancing its bioactive properties. The antioxidant power of this compound are reported in this slide, and we show that the compost tea extracted by bell pepper show whether higher antioxidant activity compared to compost tea by citrus, and this data is confirmed by the Follicle-2 test. In this case, the compost tea by citrus, which lend lower LD oxidation ratios, suggest a lower degree of lignin degradation, which explains its relative lower antioxidant capacities and limited detection of free phenolic compound. The test of antimicrobial power confirmed that there is a strong relationship between the chemical composition and the bioactivity of this compound, because in this test, the high concentration of polyphenol in CBT compost tea extracted by bell pepper correlated with the stronger antimicrobial activity of this material. Conversely, the compost tea by citrus explain lower antibacterial effect related to realized oxidized lignin content in this compound. The key finding of this work is that the compost tea by bell pepper show a strong antimicrobial efficacy against Staphylococcus aureus and Enterococcus fetalis. And this particular information that is that this material highlights antimicrobial effective efficacy against gram-positive bacteria strain compared to gram-negative bacteria strain. And this result show confirmation the higher minimum individual quantity survival, suggesting a lower susceptibility. But what is the duration of this work? In literature, there are a lot of work that explains the nucleic acid extracted by commercial compound show antiviral activity. But in this case, we discuss the application of an extra solution extracted by green compost and two different sources of green compost. The first experiment, in this case, the compost tea inhibits the envelope with zinc fraction. In fact, in this experiment, we will assess the antiviral potential against two enveloped virus from different family. The first one is the ASV1, is a linear double-stranded DNA virus from the herpeside family. To verify the Newberg effect was correlated to the viral genotype, the second virus to show for this application was an RNA virus called RSV. It's prolonged because a negative single-strand non-segmenting RNA genome surrounded by helicate, capsid, and lipid layer. In this case, we show that the co-attachment to virus cells with the different compost tea reduce the concentration of this viral. The second point of our application is the confirm the effect of compost tea on infection phases. Compost tea work the best in pre-treatment phase because the virus entered the host cell, but no significant activity was observed during the post-treatment phase, suggesting that compost tea block viral infection in early stage, lacked by target viral envelopment. So compost tea does not show any activity in cell pretreatment and post-treatment experimental condition, imply that no effect during the amplification or cell to spread. These results show that compost tea different during the extracellular infection phase targeted the viral envelopment, confirmed the idea of the next experimentation. But what is the molecular activity and pathway is involved in compost tea that stopped the antiviral activity of this viral that we use? The most common hypothesis that with the right group of phenolic interaction with the positive change domain and development of glycopyrin. In this case, the compost tea did not have a negative infection. In fact, to validate the compost tea target was the viral envelopment, we tested them against PV1 virus. PV1 virus there and non-developing virus because as a positive strain RNA genome encapsulating only by isotracease protein coding, PV is the clouding effect of polymerase. In the summary, in conclusion, compost tea from agricultural bioactives show the significant potential and multifunctional bioactive acids. In this case, we show that compost tea extracted by bell pepper has higher oxidized lignin and polyphenol, contributing to stronger antimicrobial antiviral properties, and effectively inhibits the gram-positive bacteria symptoms, for example, Staphylococcus aureus and Enterococcus fecalis, and developer virus, making them promising precise, sustainable alternative to conventional agent. So, bioactivity is relative to molecular composition, particularly on the content of oxidizing and polyphenol. The F gram-negative in a bacterial endoenvelope will suggest a specific interaction with bacterial membrane and viral envelope. This work played an implication of a biomedical application to use the agri-food compost wastes in line with the circular economy and green biotechnology, and focusing on optimization extraction to improving bioactive formulation and testing bioefficacy for the scale therapeutic development. Thank you for the attention. Thank you the Umberto Veronesi Foundation to fund this project, and thank you the University of Naples Federico II and the collaboration with the University of La Campania Luigi Mavitelli.
Thank you very much, Dr. Verrillo, for your interesting talk. And as mentioned earlier, we will now take a few minutes for the question if are present. For the moment, there are no any question for Dr. Verrillo. So I think that we can continue with our second speaker, and if at the end there will be some question for Dr. Verrillo, maybe we can forward by email to her. And thank you very much. Now let's continue with our second speaker. Dr. Armand Diaz is a postdoc researcher at the Università Politecnica delle Marche, Ancona, Italy, with a background in food science and with international research experience in China and in Spain. Today, he will present his work on the bioactive properties of purple prickly pear peel extract in human colorectal cancer cell model, highlighting in particular its ability to induce apoptosis and inhibit cell growth, a promising example of how agro-waste can be transformed into high-value products for health application. Dr. Armand Diaz, please begin your presentation.
Good morning, everyone. Thank you so much to the committee for allowing me to this webinar, for allowing me to present my thesis work, and also thank you so much to Dr. Danila Shanchosi for her kind introduction. Before to start in the main point of my presentation, I would like to give you a small introduction about this topic in general and the reason why I choose this work and also the kind of samples. Nowadays, in the context of global environmental and food crisis, it's becoming more and more important to reach high standard levels of sustainability, particularly in terms of minimizing the environmental impact of bioproducts produced by agro-industry. Cultivation of abundant species is increasing its popularity around the world. They have a wide geographic distribution and can be found in diverse climatic conditions, including North, Central, South Africa, South America, Africa, and some Mediterranean countries such as Spain and Italy. Italy is the second largest producer around the world after Mexico. Sicily is the Italian region that produces the most prickly pear fruits, around 95%. In this region, there are several manufacturing plants that process the fruit in order to obtain the juice, and as a consequence, a variety of bioproducts are obtained, such as peel, pulp, and seed. Considering that the peel represent approximately 50% of the fruit, a huge amount of peel is discarded yearly. The valorization of these discard peels can help to reduce the impact on the environment, and at the same time can be a source of bioactive compounds with antioxidant, anti-cancer, and anti-inflammatory activity. These compounds can be polyphenols, vitamin C, betalains, etc. Cancer is considered one of the main reasons of death worldwide and negatively influence the increase in life expectancy around the world. Specifically, colorectal cancer represents the third most common cancer and the second most common cause of cancer mortality worldwide. From the last 20 years, plenty of studies emphasize the potential role exerted by the consumption of plant-based diet containing bioactive components in the prevention and treatment of cancer. Taking all of this together, the present work aims to evaluate the in vitro pro-apoptotic and anti-inflammatory, pro-apoptotic and anti-proliferative effects of purple prickly pear methanolic extract on human colon adenocarcinoma HCT116 cell line. The samples were kindly donated from Catania, and they were finally kindly donated from Catania, and the first step in this work was the phytochemical characterization of the methanolic extract. To accomplish this, we evaluate the phenolic acid and flavonoids profiles and metalines together with the antioxidant activity through different methods. Next, we carried out the cell-based assays using human colon adenocarcinoma HCT116 cell line as a model. First, we performed cell viability assay following apoptosis induction, rot generation, and oxidative stress status in the cell and cell cycle arrest. Then we carried out colony formation ability. And finally, analysis of PCR and Western blot were also performed for a better understanding of the mechanism by which the bioactive compounds release their effect. The phytochemical analysis shows that the most abundant family of compounds are betalains, followed by phenolic acid and their derivative organic acids and flavonoids. More specifically, the main individual compounds detected were betanin, dihydrofluoric acid gluconide, picetic, eucomic, and ascorbic acid, and isoramnidine. As we also can observe, the antioxidant activity was quite high compared with other fruits and similar to other reports about peer-to-peer characterization. Regarding the cell viability assay, it's important to notice that for this experiment, we evaluated colorectal and breast cancer cell lines at the beginning. As we expected, due to the high content of bioactive compounds present in the extract, the treatment significantly reduced the proliferation of colorectal and breast cancer cells in a dose and time-dependent manner. This agrees with several studies which have demonstrated that prickly pear inhibits the growth of breast, liver, and colorectal cancer cells. The same concentration range was used to evaluate the effect on non-tumorigenic cells. As we can observe in these two graphics, the extracts possess less cytotoxicity for non-malignant cells compared to tumorigenic cells. For further experiments, we focus only in colorectal cancer cell line due to some problems related with the work with SKBR3 cell line. Given the fact that apoptosis is the main mechanism used by polyphenols or bioactive compounds in general to release their effect against cancer development and progression, we evaluate the apoptosis induction after treatment. As we can observe in this graphic in the left side, the treatment significantly increased the apoptotic population cells, as we see in the green bar, in a dose-dependent manner. Notice that a late apoptosis represented by yellow points in the photo in the right was significantly increased at high concentration, and they are included in the dead cells in the graphic in red bars. Further experiments were conducted to delve into the molecular mechanism underlying the apoptosis induction. In this line, from a molecular point of view, the tumor suppressor protein p53 was upregulated at protein levels. Besides, several genes related to intrinsic apoptotic pathway were modulated. For instance, pro-apoptotic caspase 9 and cytochrome C were upregulated. Meanwhile, the anti-apoptotic BCL2 was downregulated. In addition, a substantial accumulation of cells at the sub-G1 phase was observed, which is in agreement with the apoptotic assay. Related to the cell cycle arrest, the mRNA expression of several important genes in G1 phase transition were modulated. For example, cyclin D1 and CDK4 were down regulated. Meanwhile, the cyclin-dependent kinase inhibitor P21 was up regulated. Well, it's well known that reactive oxygen species can both trigger P53 activation and enhance its pro-apoptotic function, often through a positive feedback loop involving mitochondrial damage and the release of cytochrome C. In our experiment, as you can see from the graphic, the treatment increased the ROS levels in a dose-dependent manner. As I explained before, this could be one of the reasons for the activation of pro-apoptotic signaling and also cell cycle arrest. To explain more in detail this aspect, we first assume that the extract has a pro-oxidant action and induces DNA damage. This triggered a ROS level increased, and as a consequence, tumor suppressor protein P53 was stabilized and activated. As a consequence, P53 transcription factor activates several pro-apoptotic genes, such as BAXA, to induce apoptosis. Also, P53 activation could induce transcription of the cyclin-dependent kinase inhibitor P21, and as a consequence, the cyclin D1 CDK4 complex was inhibited, and the cell cycle stopped in G1 phase. Finding some explanations, some explanation for ROS levels increasing, we observed that the mRNA expression of a very important sensor and regulator of oxidative stress, NRF2, was suppressed, and as a consequence, hypothetically, the expression of glutathione peroxidase was down-regulated. Notice that no effect was observed on SODO expression. In the same line, but regarding the antioxidant activity assays, the extract decreased the activity of glutathione reductase and glutathione transferases. Meanwhile, no effect was observed in catalase or SOD activity. Furthermore, in oxidative stress, ROS are continuously produced and cause DNA, lipid, and protein damage, which leads to cancer cell death. We can observe that the marker of lipid peroxidation, T-bars, increased its concentration. Meanwhile, GSH, a very important antioxidant in cells, decreased its production. Epidermal growth factor receptor and its activated downstream signaling pathways play a crucial role in colorectal cancer development and progression. Usually, EGFR is overexpressed in cancer. As shown in this slide, treatment suppress the expression levels of EGFR in a dose-dependent manner. Similarly, HER2 expression also decreased after the treatment. In addition, PI3K, AKT, mTOR pathway was also affected. Regarding this, the extract suppressed the mRNA expression of PI3K and its downstream signaling mTOR. In addition, Western blot also confirmed that the protein levels of PI3K and PAKT were reduced. This result in general confirmed the anti-proliferative effect of the extract and also the connection between the different pathways. Further, the anti-proliferative action was also confirmed due to the upregulation of PP38 at protein levels. P38 plays an important role in the induction of apoptosis and cell cycle arrest. Notice that also ERK1/2 suppression was observed. The inhibition of this protein expression is related to reduction of tumor occurrence and proliferation. On the other hand, the protein levels of ER stress-associated molecule XBP1 increased after treatment. Finally, colony formation capability was also observed. As we see, the results show that the treatment decreased colony formation in a dose-dependent manner. This means that the extract reduced the ability of cells to undergo unlimited divisions. As a conclusion, we can say that in summary, this study highlights that pulper peer exhibit potent anti-proliferative and pro-apoptotic effects against human colorectal cancer, particularly HCT116 cells. Considering the result obtaining this work, developing effective strategies to valorize pre-peel waste can be considered as a sort of products that could have a high value despite previously being a waste product. Therefore, it would reduce the environmental waste. These interesting results highlighted the potential use of discard pre-peel as a nutraceutical or functional ingredients. At the end of my presentation, I would like to thank all of you for attention and a special thanks to my team in our lab, especially to our supervisor, Professor Mauricio Batino. Also special thanks to Dottoressa Francesca Giampieri and Danila Sanchosi, and also to all my colleagues that without their support, I could not graduate and get my PhD degree. So thank you so much for your attention, and at the end, feel free to ask me if you have any doubt or something. Thank you so much.
Thank you very much, Dr. Ahmad Diaz, for your very interesting presentation and your work. Let's now move to our final talk, Dr. Raffaelli. Dr. Raffaelli is a postdoc researcher from Università Politecnica delle Marche, Ancona, Italy, specialized in agri-food innovation and water management strategies with international expertise in Spain. He will discuss today how different irrigation strategies affect strawberry yield, nutritional quality, and the concentration of biofunctional compounds, highlighting genotype-dependent responses to water deficit and the potential for breeding more resilient, high-quality cultivar. Dr. Raffaelli, you may start your talk.
So thank you, Danila, for the introduction. Today I would like to share with you some interesting results from our experimentation. In fact, I come from one breeding group from Università Politecnica delle Marche, and we work in the improvement of selection of new genotypes of different type of plants, fruity plants, and with specific properties, with specific importance for strawberries, so Fragaria per ananas genotype. Generally, to introduce a little bit the argument, the topic that is not so well known, we start from a controlled pollination. So it means that we are checking for parental male material, so pollen, that could be useful to be crossed to an ovary from a female plant. Then we will match the plants, we create the cross, and we will evaluate in the year after from the seedlings we obtain from the fruits we obtain from the previous control cross, the progeny. And then finally, we collect the fruits from the F1 generation and we evaluate many characteristics, many features, in which we can find also the nutraceutical screening. So this is another simple scheme of what we are doing, more in details. So you can see by left to right the obtainment of the seeds, so of the first progeny from the control cross. And then the last part that you can see below, the seedlings in the field, so the evaluation from the field until the final HPLC and spectrophotometer analysis with the analysis of the nutrients effectively present in the strawberries obtained from the new genotypes. And then finally the CPVO, so it means the plant community from European Union that could certify our plant varieties that from selection, if they are effectively so good for the market, so proper for the market, they could be licensed and registered in the European and international market as new varieties. Let's move a little bit into the topics of the experiment. In fact, why we decided to work with strawberries, because they fast grown, but obviously for their also nutritional quality, because they are really important. We are agronomists, all of us, but we are also collaborating with the medicine faculty. So we are aiming. Our objective is to produce fruits that could be useful for the maintenance of a good health of the people and the normal consumer. So we are not only concentrated on the production, but also in the hidden quality. Let me use this term. And another big issue that all the people working in the agricultural sector knows that the agriculture is a big consumer of water. Actually, in the last 20 years, many research topics were focusing on the finding on new solution to optimize the water consumption. In fact, if you can see the histogram that I show you in this slide, you can see immediately that the agriculture is counting more than the other sector, human sector, for the at least 70% of the annual total water withdrawal. So it means that the main part of the water is used for irrigation. So we tried to combine all these two big topics, the health quality of the strawberries and the water consumption problem of agriculture, and we created this experimentation that you can find also in the Agronomy and DPI journal. What we did. We start at the beginning with two specific objectives. We can see also three, but first of all was from our huge population that we have in our field, we have many selection, cultivars and so on, which one was more adaptable for a soilless condition, so it means grown in a soilless environment, and could maintain the highest quality in terms of nutritional, the yield and organoleptic quality of the fruits, could maintain all these aspects also in low shortage condition. So it means with reduced irrigation condition, which is the best. This is the key point. Secondary, we would like to share our model, creating effectively model to be used, to be implemented not only in our research breeding program, but also in other plant breeding programs, because we think that this is so useful, so important, so crucial for the creation of new selection, appetizable for the market in terms of this condition, climate change and all the problems connecting to the different rainfall patterns and the lack of water, and maintaining the quality of the products. And secondary, we would like to study a little bit which are the influence of the irrigation in the nutritional composition of the fruits, because we are, as I told you, agronomists, but we are interested also in the evolution of the biomolecules, biofunctional molecules present effectively in our food. So we start with selection of three different plant genotypes. We have two cultivars, so just present on the market, that are called Lauretta, Francesca, and one selection that you can recognize by the number, because it means that it is not actually on the market, so it's not registered through the CPVO, as I told you before, the plant communities from European Union. And we work on these three plants. Why? Because they show in the field, so in a normal field during these years, great capacity of production and also a little bit of with high nutritional quality and so on. So we would like to select these three in particular the cultivars, and to test them at different irrigation stages, different irrigation supplies. We create with some specific pots a specific substrate that in reality is from a really famous brand of substrate. We can't say the name, but is really proper for berry fruits and is composed of peat, silicate, coconut fiber, and ecofiber. And we decided to analyze that substrate from a specific private lab, knowing all the characteristic features, all the features, physical, chemical features of this substrate. So like the pH values, the porosity, to know effectively how the hydraulic mechanism could be occurred inside this substrate, how much water is possible to retain in this specific substrate, knowing all as possible of this product. Then we decided in our experimental greenhouse to divide in 3 different blocks, 3 different big tables, probably adapted for the place of these plants. And we divided 3 different tables depending on the irrigation management we would like to perform. So we have 100 field capacity. It means the 100% of the saturation of the substrate I told you before. So it means that the plant has continuously water available for its absorption. A lack of 25% from the 100%. So we create the second block, WS1, we call it. And finally, in something like drought condition, according to the specific characteristic of the substrate, we are less than 60% of the total water capacity. So it means that the plants were really, really in a drought condition in that situation. And I would like to tell you also that all these plants were replicated. So we have totally 90 plants, 30 per each genotypes. Every color represents a specific of these 3 genotypes. I would like also to tell you that this experiment was done in 2022 and replicated in 2023. So all these data I will show you are averaged in 2 years. First of all, what we analyzed. I can show you obviously all the data. We analyzed the water use efficiency. That is a specific agronomical measurement that could give you the capacity of production of a specific plant based on the water we provide to the plants. So we obtain the yield in terms of kilogram of fruits obtained from 1 cubic meter of water. So it means with 1,000 liters of water, how much is the yield of the plants treated at this condition. So on the left, you can see immediately the soil moisture sensors during all these experimentation that we take, we detect through specific sensors. So 100%, the second treatment and the third treatment with 3 different colors. And on the right, the water use efficiency. And we know immediately that you can see in the 3 different histograms, block of histograms, the 3 different genotypes, that the second treatment was practically the best, with around, for the selection and cultivar Lauretta, around 30 and 34 kilograms every cubic meter of water. So it means that is the best treatment in that case, if compared also with the full water treatment. We can say, okay, that these are 2 the best. The Francesca is not appetizable. In this case, I can say that, then I will show you also the nutritional analysis, but for an agronomical point of view, Francesca is not affordable. It's not possible to take him away and to keep it, to maintain it for a specific agronomical schedule for farmers. It's not adaptable. Then we analyze the organoleptic quality. So I decided to show you simply the treatatable acidity and soluble solids of fruits. We know immediately that the citric acid concentration responds in a proportional way to the drought induced in the plants. So it means that the higher is the concentration of the drought induction, higher is the concentration of citric acid, like a small response of the plants to this condition. Instead of for the soluble solid that we simply take through a refractometer, seem to be not so well dependent by the water intake, but at least a little bit stable in some way, in some in this condition, not with valuable differences among the different treatments. And finally, as I told you before, we decided to analyze all this fruit for these 2 years with 6 different type of analysis. First of all, we analyze with screening, general screening, the total antioxidant capacity and the total phenolic compounds with Trolox equivalent antioxidant capacity analysis and Folin Chocolate analysis for the total phenolics. And then through a specific high-performance liquid chromatography, we decided to analyze the ascorbic acid, also known as vitamin C, folates, phenolic acid, and anthocyanins. Here you can see immediately a big scheme of the plants with their every histogram represent a specific treatment with the content of the specific molecule we are analyzing. Then I show you better. In the green one, you can find the folate. And we analyze the folate, as I told you before, through HPLC. Is a derivative, as folic acid. It's possible to find this molecule, also known as vitamin B9, in the plants, and is useful, for the nucleic acids structuring and other types of biochemical pathways inside our body. Then we can find the vitamin C, so ascorbic acid, useful as is radicalic scavenging activity and with the collagen production and so on. Then all the phenolic acid. In their case, we analyze the chlorogenic acid, the lactic acid, and caffeic acid. Then the anthocyanins that we know are real flavonoids, real present inside the strawberries, inside the fruity species, with a specific, obviously, detection of pelargoldin 3-glucoside. That's the highest quantity of anthocyanins effectively present inside the fruits, this type of fruits. Then finally, the spectrophotometer with the Trolox equivalent antioxidant capacity and the total phenolics. We created this nutritional content, like a nutritional score, let me use this term, to average this data, but obviously this content is not a specific unit of measurement. We know that all the molecules as calculated as milligrams per 100 grams of fruit weight, instead of the folate that is really few concentrated inside the fruity plants is calculated as micrograms. So we have to discuss about it. We know immediately that Francesca and Lauretta were pretty much the best. Probably they were registered on the market thanks to this capacity of nutritional content. For that reason, they were probably released in the past. The selection was not enough, but removing the folate content, because it's a little bit, is not so correct to put there because it's in microgram. We put only to check it out effectively, the content of this particular molecule. We have to detect that Francesca and Lauretta were really, really competitive among them. So effectively, actually, after all these analysis, we know that actually Lauretta could be the best genotype in that case, because it's productive, like the selection, and also highly full of nutrients, like Francesca, that is not productive. So we can use Lauretta for future crosses in the next years. Some further observations, as I told you before, we know immediately that anthocyanins content and total phenolics were really, really reactive to the growth induced. So we know that we can use probably these molecules as growth indicators. Maybe it could be useful also for some specific biomarkers in the future to improving the breeding with new type of genetic breeding, molecular breeding. They could be a source, probably. And finally, I create a correlation map. This is more to help effectively the breeders in their private company, because all these nutritional analyses are really, really expensive to keep it away. Also, because we were working with a lot of selections. So if you want to adapt, you have to do an economical choice. I suggest, thanks to this correlation map, personal correlation map, that the total phenolics was one of the most reliable analysis. You can see that the redness is corresponding to the effectively correspondence among the analysis. So we know that total phenolics, instead of for pelargonidin 3-glucoside, is really, really sensible, is really related to the other components, nutritional components, as we saw. So let me introduce the conclusions. We know that Laureta represents one of the most resilient genotypes from UniVPM breeding program, so is adaptable for new crosses to create new varieties of plants, adaptable for drought conditions, so with low water intake. We know that this experimentation was really useful for the choice of the water treatment, because we know that reducing the water, the normal supply of water of 25%, was pretty much the best to improve also the quantity of the production. So it doesn't mean that more is the water, higher is the quantity of the yield. Finally, we know that total phenolics and anthocyanin are really reactive molecules, and so they can be used as growth indicators to be adaptable and to be studied more also in a molecular point of view. So finally, we can say that creating a new plant selection model is practically possible as shareable with all the people, and is important that we that are working in research are sharing these models to improve also the private sectors. So thanks for the attention. Here you can see my LinkedIn, and for any question, I'm free to respond to you. Thank you.
Thank you very much, Dr. Raffaelli, for sharing with us these valuable results. Now we will open the Q&A session. And please, if you haven't already done so, write your question in the panel of Q&A now. I can see that there is one question for Dr. Armas Diaz, and they ask to you, what was the bioavailability of betaline?
Okay. Can you hear me? See? Can you hear me? It's okay?
Yes.
Yeah. Okay. Okay. Thank you so much for the question, and I think is a very important question, and will help me to explain a little bit something that I didn't say during my presentation. Okay. The person asked me the bioavailability of betaline, which are the main bioactive compounds in my extract. And maybe all of you noticed that in my presentation, I have 3 treatments with a whole extract. And then because of when I did the composition, the betalines, as I expected, were the highest family. But the difference between betalines in general as a family and the next one, the polyphenols, was very high. So at that time, I expected that the bioactivity of my extract could be mainly related to betalines. And then I decided to make, let's say, like as a positive control, one to simulate to have the same amount of betaline, specifically betanin, because betaline, there are 2 families, beta-cyanins and beta-xanthines. But in the red variety, the majority are beta-cyanins, specifically betanin. So taking all of this, I decided to make like a positive control using the same a similar concentration of betanin in my extract. But interesting, the results show me that at the same concentration, I didn't see any effect for betanin. So after that, I think that the effect of my extract was not related only or directly to the betaline contents, was more synergistic effect. But of course, I studied first everything related with the betaline family, and in general, I can say that betaline, they have let's say low bioavailability. But also in our case, I couldn't do this work because I didn't have time for some reasons. But we usually in the lab, we used to work with raw material and also with digested material. Even if we work in vitro, But we always try to work with the raw material and the digested material to see the bioavailability. In this case, as I said, I couldn't do this study for several reasons, but in general, not in my time because I didn't do. We used to do in the lab, but in this case, I couldn't. The bioavailability is low, but taking into account that the concentration usually is very high, it can be absorbed. But in this case, for my study, I didn't show this. But in general, what I read was that the beta lines bioavailability is low between the family. Usually, in the beta xanthine have a very high bioavailability compared with beta cyanins. And in my case, it's reaching beta cyanins, but for my study, I cannot reply directly the answer, but in general, it's low.
Thank you.
I hope
Thank
that
you very much.
I was able enough to I convinced the person that asked.
Thank you very much for your answer. And I can't see any other question. So we are reaching to the end of today's webinar. My sincere thanks to Applied Science, to MDPI for hosting this webinar, to our three speakers for their excellent contribution, and to all of the audience. I hope this session has provided valuable insight into how nutrition, agri-food byproducts, and biotechnological innovation can work together to promote both human health and environmental sustainability. And so thank you very much, and I look forward to seeing you in future events.
I'm sorry to interrupt. There is one more question in the Q&A box.
Okay. Now
it's the same question, but I don't know. The person has some problem in the audio. I don't know if you hear
yes.
me.
There is the same person that asked if he can repeat because she can't hear.
I don't know.
I think it's her problem, not
Is it because
in
if
the
you
sense
hear
that
me, you listen to me, I don't know.
Yes. Maybe
I
she
can repeat
can.
if I have to. It's not a problem, but
Maybe she can also receive the webinar. My research is on. That's why. Okay, she said that she also worked with prickly pear plants.
yeah, but the problem is if I don't know if I still continue talking or I reply again, I don't know if she or here, I don't know, listen to me now. So I don't know because if I repeat or something, I don't know if she listen because she doesn't hear or doesn't say anything. So I don't know what to do.
I think that she can see the answer in the webinar that will be very soon in
Okay.
the website, because all of us as hear the answer of Dr. Armas Diaz. So I think it's a technical problem. So I think that she
Well, I don't know if she can find my email or something. She would like to write me
maybe you can write in chat your email.
Okay. I write my email. No, I think she can find in the presentation any information. And she contact me because I don't know now where to write my email.
Okay. She wrote her email, or he, I don't know, wrote her email in chat. Maybe you can contact him, her.
Okay.
Thank you.
Thank you so much.
Dr. Canciosi, do you maybe have anything else to add?
For me, it was a pleasure to participate to this webinar. And so, like I said before, I thank all of you and all the participants to this webinar.
Then thank you all, Dr. Canciosi, Dr. Diaz, Dr. Raffaele, and Dr. Verillo for all your time and interesting contributions to this topic. I would also like to thank our participants as well. I hope you found this webinar both interesting and insightful. Please note that certificates of attendance will be made available shortly after all registered participants on SciForum account. If you have any questions related to this webinar, simply send us an email with the webinar date and title to journal.webinar@mdpi.com. As a reminder, the recording of the webinar would be made available as soon as possible on the webinar webpage. We would greatly appreciate if you could take a couple of minutes to complete our satisfaction survey by following the link provided in the chat box. Your feedback is essential and important for us to improve future webinars. I wish everyone a nice day, morning, evening, wherever you are in the world. Thank you again and see you another time.